Thank you, Bunny. I'm going to ask you to answer questions in the chat so that we can move on to our next speaker, who is Terry Jones, who will be speaking to us from Charité University in Berlin about genetic variability, including a gene duplication in the 2022 global monkeypox outbreak. That looks good, Terry. Thank you.
Great, thank you. Okay, well, I'm going to assume that you've read the title, and I'll save myself 30 seconds by skipping to the next slide, which is by way of general introduction. I extend warm greetings from Berlin, where I work at Charité, which is a large university research hospital. It's been around for about 300 years. We have 15,000 employees, about a third of them are involved in research, 3,500 hospital beds, and we have about 15,000 beds that are covered by diagnostics. And I work at the Institute of Virology with about 100 other people. I want to make a few preliminary comments about orthopoxvirus terminal genes. These typically modulate host range and immune response, and can impact things like transmission and virulence and so on, as we heard a little bit earlier. And it's well accepted that changes in these genes are associated with the narrowing of host range, which gives the virus a chance to specialize and will change selection pressure, maybe increase it, maybe decrease it, and lots of different things can happen. And also changes in these regions are well documented on short and also long timescales, and we've already heard a little bit about this today. I want to mention 3 papers very quickly before I go on. I'm going to refer to these in the talk. One is this paper, which is about the role of gene loss. The last author of the paper is actually in the mini symposium with us, and we will hear from him today. That's Elliot Lefkowitz. That paper is great. It's got a lot to do with host range and gene loss, and it has a wonderful supplementary table, which is a huge Excel file, which people will find useful if you're doing bioinformatics on poxviruses. The next one is a paper that talks about this accordion-like expansion and contraction of the genome, which is reminiscent of bacteria. Also a very interesting paper, possibly quite relevant. And this is an example of how you can get this kind of change to happen in the short term, just in the space of a few passages in cell culture. And finally, there's a recent paper which tries to make sense of the chaos of poxvirus genes and their names and their possible annotations and functions that just came out last year. Okay, so here is a figure. This is figure 8 from the first paper that I just mentioned, and you can see this is the genome layout of a bunch of orthopoxviruses. This is the left side, the right side, and the middle, and each row corresponds to a particular orthopoxvirus species. And what I want you to see is that the red squares and the yellow squares and the black frame squares, these all represent some form of gene inactivation or stop codons or deletion and so on, or disruption of some form. And this highlights the pattern of changes in the terminal genes in a whole range of orthopoxvirus species. Also, we had a paper a couple of years ago about variola virus, which is the cause of smallpox. And without going into details here, in the top row, you can see a bunch of open circles. These represent intact genes in cowpox, like orthopoxviruses. And you can see that during the course of evolution, by the time you get to the modern variola virus, all of these circles in blue indicate genes that have been inactivated one way or another. So this is a pattern of what happened in variola. And you can make a plot of the number of genes that have gotten lost over time. Some of these sequences are from Vikings and from slightly younger sequences. So starting from about 600 AD or 600 in the Common Era, variola had lost about 40 genes. 300 years later, it had lost about 47. And as of about 300 years ago, about 52 genes had been lost. I'm not trying to suggest that this happens in a linear fashion, because it probably doesn't, but you can see the general pattern of gene loss over time. And this is obviously on a much longer timescale than 3 or 4 passages in a cell culture. So what should we expect from the human monkeypox outbreak? First of all, one comment which is kind of obvious is that monkeypox in humans is never transmitted in this fashion before. In fact, the longest chains that were known were from strains that were Central African, and they were of length of about 7. So the virus is finding itself in sort of unprecedented territory with the opportunity to specialize and adapt to human hosts. And as I mentioned, these genome changes have occurred previously in humans, and they have produced viable viruses. We have quite good evidence of that. I'm going to mention 3 of them a little bit later. And we had a case like this in Berlin, which is the subject of the rest of this talk. Assuming that human-to-human transmission goes on, these changes will continue to appear, and the adaptation may play out over a period of years or decades or even centuries if the virus manages to remain in humans. I think it's safe to say that they will happen. I don't think there's really any doubt about that. So we had a case, or we had a series of cases starting also in May of this year, and we saw the kinds of things that everyone else was seeing in the genomes, some of the things that have already been mentioned. But then we had one particular patient, and when we sequenced the virus and aligned the reads against the reference sequence, there was this big empty region that looked like a gap. And of course, that's very provocative because if you think about it, it's obviously not due to low coverage because the coverage is actually very good in this particular sample. So it looks like a gap, but it's not a gap. It's a reference mismatch. And so in order to figure out what was going on, we designed careful PCR primers that would span the region, did PCR, and then did Sanger sequencing to figure out what was going on. When I say careful PCRs, I mean PCRs with primers that won't match elsewhere. At least the forward primer will not match elsewhere in the genome. And this revealed that a region from the beginning of the genome on the left of 856 nucleotides had been duplicated to the right-hand end of the genome. And this included D2L, which is a gene or possibly a gene. And also in the process, 2 other genes had become truncated and 2 were deleted. And there's the GISAID ID of the sequence. So we go from this green thing on the left to this circle on the right. So this raises many questions immediately, and I've got some answers here on the slide as well, so I don't keep you in suspense, and I keep my talk to 12 minutes. First of all, is this a viable virus? You know, is this a mutant that maybe couldn't replicate or had other problems and was not representative of a virus that was still infectious and so on? And the quick way of answering that, of course, is to try and isolate it, which we did. And that was successful, and that was done in Vero E6 cells more than once. Then, of course, we also were curious to know whether this particular virus variant or mutant was present in other lesions, and it was. And of course, after we did the isolation, we did the sequencing to try and confirm that the duplication was still there. And that also gives us another yes on this slide. So you get the pattern. So at that point, we thought, okay, this is really something. We have a viable virus. It's obviously managed to spread through the body. And then we asked the obvious questions. Is there a difference in growth kinetics that we can see? The answer is yes. I'll come to the details. Is there a difference in host protein expression? The answer is also yes. And I'll show you a little bit about that. And then 2 more questions. Do we see this change or changes of this type in other monkeypox virus sequences? The answer again is yes. You won't be surprised to hear that because we've already seen some presentations. And then finally, to disappoint you all, can we explain the differences and do we know what the 5 genes involved actually do with any confidence? And I'm not sure that question will ever be answered, but we can try. So just to show you a little bit about the replication kinetics, we evaluated the growth in 7 cell lines, and we have more that we can use, obviously, but we immediately saw that there was a replication advantage in HeLa cells. It's not a big one, but it's like a twofold advantage, sometimes a bit bigger depending on things. This is with and without interferon pre-treatment. And also in 4 other cell lines, we also saw a similar growth advantage. In the Vero E6, it looks like maybe there's an advantage after only 72 hours. And there were 3 cell lines where we didn't see any advantage. I can show you a bigger version of this image if you like in a minute, but I'm trying to move quickly. We looked at host protein expression levels using Western blot analysis, and we found 3 host proteins that have reduced levels. These are GRP78, which is it's a protein that can help to detect unfolded proteins as a result of ER stress, and it helps to maybe get the proteins properly folded or to get rid of them. Then there's a reduced level of this H2AX protein, and there's this particular phosphorylated form, this gamma form, which occurs as a result of double-stranded DNA breaks or detection of that. And there's a reduced level of caspase-3, as far as we can figure out, which obviously is likely to have something to do with apoptosis. This is all research in progress. This stuff is going on in our S3 and other laboratories on a daily basis. So I can't give you, final conclusions about any of this, but there's definitely growth differences and there's definitely protein differences. So going back to my earlier question about do we see changes elsewhere that are similar, there's 3 that I think are worth pointing out. The first one is there were 2 patients that were found in the South Sudan in 2005. This also was mentioned earlier. In this case, there were 12 genes from the beginning of the genome that got duplicated to the end. And that includes this D2L gene and 11 others. And it knocks out the 4 genes, the identical 4 genes at the end. It's not known what the significance of this is. The next one is some sequences that were initially from West Africa dating, the first one, 1958, is the Denmark case. But most of these sequences are West African, and they also include the US 2003 outbreak sequences. We noticed that there's a duplication from the right-hand end of the genome back to the beginning of these same 4 genes. And it's very interesting because if you make a phylogenetic tree that only includes the central region of the genome, which is the more conserved region, and you leave out the ends of the genome, which is where these genes are, these 20 sequences which we have, they still form a monophyletic clade. And in fact, if you look at the next strain, monkeypox build of all of monkeypox sequences, not just the humans, they have 26 strains in there, 26 sequences, and these sequences are in there. So this is an indication that this duplication happened probably a long time ago and has been circulating probably ever since 1958 in one species or another, which is obviously is telling us that this is a fit, it's a fit variant, or it's a fit genomic change that is circulating with genomes that don't have this change. And finally, there's about 5 patients that were identified in Central African strains that have a deletion of a couple of these final genes that is suggested to increase fitness in humans, where fitness, I think in this case, would have to be understood as maybe attenuating the virus, maybe making people less sick and resulting in more circulation and maybe more transmission, because it's knocking out genes that would normally antagonize immune system functionality. Although with poxviruses, who knows? Okay, just quickly, the mystery is about what do these genes actually do? These things are very unclear. Most of what is known comes from vaccinia and studies in mice or cell cultures and so on. And as has been pointed out, including in the paper that I mentioned earlier, that has Elliot Lefkowitz as the first author, the situation with poxvirus genes and the annotations and so on is a bit of a chaos, and it's very hard to make sense of. 2 of these genes seem to be quite reasonably well understood as to what they do, at least in vaccinia and cowpox. And I'm not going to go into the details. You can have a look. You can look them up later and try and solve the puzzle yourself. These things could be expected to possibly attenuate the virus. And then we have the duplication of this first gene. If it is a gene, it's just 63 amino acids. In some cases, it's not annotated at all. So some people don't even think it's worth mentioning. There's annotations that seem questionable. You'll notice that there's an annotation of C16L and B22R, which is similar to one of the genes which is knocked out. So, it could be that nothing really has changed. And then in the recent paper that I mentioned, there's a suggestion that there's a BCL2 fold, which could imply some role in inhibiting apoptosis, but we don't really know.
Terry,
Okay,
apologies for interrupting, but we're just at time, so if you could just wrap up quickly.
Okay, it's perfect. So I have a final slide, which is acknowledgements. There's a dozen people at our institute who have been working on this since May, very intensively in most cases, in the S3 lab, in other labs, doing sequencing, doing bioinformatics, and so on and so forth. We have a really great team. And we have 3 collaborators from other departments or from another hospital who have provided us with the samples. And thanks for listening. I'm happy to answer questions, or at least try to.
Thank you. I think we'll have to do the questions in the chat. That was a fantastic talk, and I have no doubt you'll have many questions.